Thank you, Marcy, for the invitation. Good afternoon, everyone. Sorry. My name is Baharan, and I'm a PhD student in the Chemical Engineering Department. And I'm presenting today a machine learning-based method for metabolic flux analysis of patient tumors. So my today's presentation is a part of our work in Deepak Nagra's lab in collaboration with Dr. Daniel Wall's lab. Glioblastoma is the most common and the most aggressive brain cancer with the 5-year survival rate of 6.9% and average survival rate of 15 months. GBMs account for half of primary brain tumors, and they recur frequently in 80% of cases. On the left, you can see the survival rate of different brain cancer, and you can see that glioblastoma or GBM has the worst survival rate among other brain cancers. Metabolic reprogramming is a hallmark of cancer. And in general, we can define metabolism as a set of chemical reactions that occur inside of the cell. And these reaction includes converting food or nutrients into energy and macromolecules like RNA and DNA. Metabolism supports cell growth and other cell functions. In cancer, cancer cells accumulate metabolic alterations that allow them to gain access to conventional and unconventional nutrient sources, utilize these nutrients toward the creation of new biomass to sustain deregulated proliferation, and take advantage of metabolic regulation on the cancer cells itself, as well as on the other cell types in the tumor microenvironment. They take advantage of all these metabolic regulation for their proliferation. Although metabolic rewiring is beneficial for cancer cells, if we understand cancer metabolism, it's a targetable liability with a therapeutic window. 13C glucose tracer is a common way to study metabolism, and you can think of metabolism as a set of stir tank reactors that are connected together, and there is a flow between the tanks. And if we add some ink to one tank based on the flow rates, the other tanks will be colored with some concentrations of ink. And if we measure the concentration of ink in different tanks, we can write balances for the ink concentration, and we can calculate the flow rates between these tanks. So this is a concept in chemical engineering. And this is very similar when we use the 13C glucose tracer. So glucose at first it had 6 carbons, and the atomic mass of carbon is 12. And when we use the tracer, instead of having carbon with 12 atomic mass, we have it with 13 atomic mass. So the 13C glucose tracer has 6 more mass unit than the natural glucose. So when we use a tracer, this can be like ink. It can be spread into the downstream metabolites, and we can see different patterns in by running into different reactions. And each of the patterns, we call it a mass isotopomer. So each pattern has 13C carbon in one of the positions of the molecule, and we can group these isotopomers into mass isotopologues, where we have like similar number of 13C carbon in one group. So this would be a mass isotopologue. And with the mass spectrometry, because these metabolites are heavier than the normal metabolite, It can be measured with mass spectrometry, and we can find a distribution of these mass isotopologues. So we use this technique in our study to find the metabolic rewiring in GBM patients. And to do that, we have some patients and we infuse them with 13C glucose. This infusion was during the surgery, and after the surgery, we had 3 samples from each tumor, enhancing, non-enhancing, and adjacent cortex. And this classification is based on the histology of these samples. So enhancing and non-enhancing are both tumor. Cortex is normal tissue. The difference between enhancing and non-enhancing is based on the penetration of blood vessels into the tissues. So here you can see that this part is a blood vessel, so enhancing has more penetration of blood vessels. We have the 13C tracing in these tissues, and we can measure the metabolites from different samples. And then also we have the GBM grow in the PDX or patient-derived xenograft mouse models. And again, we have the 13C glucose tracing in mice. And after the infusion, we have the tumor, cortex, and some plasma samples from the PDXs, and then again, we measure the metabolites in mice. So glucose is metabolized through glycolysis, this part, and pentose phosphate pathway, and its carbons are utilized in many downstream metabolites, as you can see. Tracking 13C incorporation into downstream metabolites allow us to understand the more active pathway by comparing the metabolites in glioma and cortex tissues. So here for the glycolytic intermediates, we saw a similar enrichment for the mass isotopologue distribution between the cortex and the tumor samples. We saw pyruvate and lactate have higher enrichment than other glycolytic intermediates, and we think that that lactate might be taken up by cortex and GBMs both like at the same rate. With this, we can conclude that glycolysis is an important pathway in both GBM and cortex because we don't see any difference between the glycolytic intermediates and enrichments in these tissues. Then we looked at the TCA cycle intermediates, and this is important because TCA cycle is a hub that allows for both the oxidation of glucose-derived carbons and their conversion into other molecules such as neurotransmitters and amino acids. So when we looked at the TCA cycle intermediates, we saw a decrease in enrichment of these metabolites in the tumor samples. And this means that the TCA cycle activity is reduced in the GBM than cortex. And when we looked at GABA enrichment, which is again a neurotransmitter, we saw again a decrease in the tumor samples. And also we looked at serine enrichment in both GBM and cortex, and we saw almost similar levels of enrichment in both tissues. But when we looked at the patterns of enrichment in serine data, we could see that N+1 pattern had higher levels in the GBM, while N+3 pattern has higher enrichment in the cortex. And also we see the same thing. This is for mouse, and this is for patients. And we see, again, the similar trend in the patient data. As you can see, M+3 is higher in the cortex, and M+1 is higher in the tumor samples. So we would like to see what are the sources of serine and what is the reason that we see different patterns in enrichment in GBM and cortex. So we looked at serine in the plasma and the 3-phosphoglycerate, which is the metabolite that produce serine in de novo pathway. When we looked at the enrichment patterns in plasma, we saw that M+1, so like serine in plasma is composed of only M+1 enrichments, and the 3-phosphoglycerate is similar in both cortex and GBM, and it is composed of M+3 majorly. So from this, we hypothesized that GBM rely on serine uptake because we saw M+1 enrichment higher in GBM samples. And also we see as the plasma serine has the M+1 enrichment. And then to confirm our hypothesis, we designed another experiment with the mouse. And so here you can see the GBM patient-derived xenografts. So we grow GBM in the brain of mouse, and you can see here, this is the tumor part. And when we restrict their diet with serine and glycine, so these mouse, they have reduced levels of serine in their plasma. And you can see the tumor size reduced. Yeah, and then another metabolites that we looked at was the nucleotides in the purine metabolism. And we see here that guanosine monophosphate, guanosine diphosphate, and guanosine have, all of them have higher enrichment in the tumor samples. And this means that nucleotide synthesis is higher in the GBM. To summarize all of these results, we can see that glucose, because the enrichment of glycolytic intermediates was similar between cortex and glioma, we understand that glycolysis is important for both cortex and glioma. We saw TCA cycle intermediates have lower enrichment in the glioma, so TCA cycle activity is lower in the glioma. We saw higher enrichment of nucleotides in the glioma, so glioma needs to synthesize more nucleotides, and this is actually confirms the hypothesis that glioma cells want to proliferate and divide, and they need more nucleotides for their divisions. So they need some sources for nucleotide synthesis. One of these sources to synthesize nucleotides in de novo is serine. And by looking at the enrichment patterns in plasma, we could see that glioma cells can uptake serine from the plasma and use it toward synthesis of nucleotides. And to confirm this hypothesis, we did the experiment with serine glycine free diet, and we saw when we do not have any serine in the plasma, it will result in reduced growth and proliferation. So all of these findings are based on 13C glucose single time point experiments. However, high 13C enrichment at a single time point doesn't necessarily imply increase biosynthetic flux. So you can see here, these metabolites can be produced from different sources. And one thing we'd like to consider is that how much of these reactions can produce these, like the ratio of synthesis through different sources. And another thing that is that when we have enrichment patterns in the sources, we got enrichments in the products. But when there is an unlabeled source to produce a metabolite, the metabolite has lower enrichment. So we can say, like, if a reaction is more active in some cases, because the source, if the source is unlabeled, the product is also unlabeled, and we don't see any enrichment pattern in it, but the rate of that reaction might be high, and that reaction might be important. But another reaction might be at low rate, but the reactants have some enrichment, so the product has higher enrichment. And based on this, it can be like some bias in just by looking at the data. So we want to quantify the rate of the reactions. And one way to quantify the metabolism is to use metabolic flux analysis. And with this tool, we have already the data from our enrichments, and also we have a metabolic model consists of all the reactions. And by using these two, we can write all the mass balances on these systems and find the rate of these reactions for flux of reactions. And so for to quantify purine metabolism, we'd like to do another experiment, because purine metabolism is very slow, and during the time of surgery, the enrichment patterns are on a steady state, and we need multiple time points. If we want to write the mass balance equation, we need multiple time points to fit our data into the measured enrichments. And so for that, we again have the GBM PDXs, and we infuse the 13C glucose into them. But after that, we harvested the tumor in tumor and normal tissue at multiple time points. So the difference of this experiment is that we have multiple time points instead of just having 1 point. And then we measure the mass isotopal distributions. And here I'd like to discuss how I write the mass isotopal logs. So here we have a metabolite, guanosine monophosphate, with the enrichment pattern of N+1. So GMP can produce from IMP, inosine monophosphate, or through guanine and R5B. Guanine is non-labeled, but R5B has N+1 labeling. And GMP also consumes through these two reactions, GMP to GDP and GMP to guanosine. So when we write the mass balance on GMP N+1, it would be 2 terms producing and 2 terms consuming GMP. And on the left, we have the accumulation of GMP N+1. And so to estimate the flux of reactions, concentration of metabolites, and the MIDs of input metabolites, we solved an optimization problem. We already have the MIDs from the experiments, that's the enrichment patterns we have, and we also have the concentrations of metabolites. So we want to minimize our error to be close to our experimental values, And we have some constraint because we have a metabolic model and those reactions are connected together. So this one shows the overall mass balances on the system. And we know that the enrichments should be one for each of the metabolites. And we set a bond on flux values to be in a reasonable range. And this one, this is the equation that I showed you in the previous slide, but generalized on the for all the metabolites. And this is the time-dependent isotopic log mass balances. So to optimize this problem, we first initiate some MIDs and fluxes and concentration, all of these parameters first initialized, and then we solve the ODEs and find the MIDs and concentrations. And then we calculated the differences and find the objective value. And with the nonlinear programming, we adjust the parameters and it goes and it runs until we find a minimum for this problem. So using this method, we can quantify the fluxes of purines. And when we compare cortex and GBM, we could see that IMP is produced mostly from the salvage pathway in both cortex and GBM. The IMP to GMP, which is called de novo GMP synthesis, has higher rates in the GBM. And also we found that inosine and guanosine production is higher in the GBM, which confirms that GBMs need more nucleotides because they want to divide and proliferate. So then we want to focus on the GMP synthesis because we know there is a drug that targets enzyme inosine monophosphate dehydrogenase or IMPDH. And so we usually focus on the reactions that we have a drug for them. And here we have a drug called mycophenolate mofetil or MMF that targets the de novo GMP synthesis. The GMP can also produce from the guanine in the salvage pathway. So besides having a drug for this reaction, we also saw in our dynamic model analysis when we have a radiation in GBM and cortex, GBMs rewire their metabolism to rely more on the de novo GMP synthesis. As you can see here, after the radiation, the rate of de novo GMP synthesis goes higher, and the salvage is lower. So this is another reason that we think this drug can also be used in can be used after radiation. We also looked at different metabolites in purine metabolism in patient data, and we saw GMP is the only metabolite that has higher enrichment in the tumor samples in all of the patients. We don't see this pattern in other metabolites, and we think that GMP synthesis is remarkable here. But in the patients, we can solve the optimization problem to find the fluxes. So we don't know how much of the GMP synthesis is from the de novo synthesis and how much of it is from the salvage. So we'd like to quantify that in the patients. And the reason that we can't use conventional metabolic flux analysis in patient is that we only have one time point. And for the metabolic flux analysis, we need multiple time points for the on a steady state problem to fit our data into the measured MIDs. We can solve the MFAs with one time point if and only if it's at a steady state. But the purine metabolism is slow, and we only have one time point on a steady state. So we can't use conventional MFAs for prediction of fluxes. And we think that we can use a machine learning model to predict the fluxes in patients. And for that, we need some simulated data because we have a low number of patients and mice, and we can train the machine learning on a very limited source. So we decided to simulate some data and then train a machine learning model, and then use some experimental validation for our machine learning model. And to simulate MIDs, we generate uniformly distributed fluxes and concentrations, and the range of these distributions are from our previous study for with the metabolic flux analysis. And then we have the uniformly distributed ratio of de novo GMP synthesis to the overall GMP synthesis, and this is the value that we want to predict by our machine learning. And we also have a metabolic model. So all of these, we put it into an optimization problem, and we solve this to find the fluxes. And for each of the simulation, we find a different set of fluxes that are constrained together. And we use these flux values, and we solve a system of ODEs to find the MIDs. And so because our MIDs are on a steady state, we have the accumulation term on the left, and we find the MIDs by solving this system. And so at the end, we have the MIDs and the ratio of de novo GMP synthesis to the overall synthesis. And these are the values that we use in our machine learning model. The MIDs would be the input data, and the ratio is the output of the machine learning. So in our simulation, we have different reactions. I'd like to mention most important reactions in the glycolysis and pentose phosphate pathway, serine metabolism, and purine metabolism. We have some reactions that we found important from our previous study, like serine plasma, which we found GBMs rely mostly on serine uptake. So we add these reactions into this model. And by solving the optimization problem, we found a distribution for the fluxes. And so you can see that we first initiate this problem with the uniformly distributed fluxes, but after going into this optimization and constraint based on the mass balances, we for each of these fluxes, we got a unique distribution. And we use these fluxes and put it into the system of ODEs, and we find the MIDs. And the MIDs are at different time points. Yeah, so you can see here that we have different time points, different metabolites, and different MIDs, as you can see here. So with this simulated data, we split the data into training, test, and validation data set. And we centered and scaled data based on our training feature. We think that would be beneficial for our input data, because our MIDs are at on a steady state, and by scale and center the data, we reduce the effect of time on these MIDs. The fluxes are at a steady state, and we want to somehow be at the same condition for both fluxes and MIDs. So with this configuration at the input data, we have different metabolites and different MIDs. And we like to use a convolutional neural network to predict the ratio of de novo GMP synthesis to the overall synthesis. And the two-dimensional convolution layer, it captures the mass balances on each isotopolog. So you can see that it's a column vector. The kernel is a column vector that goes through different metabolites, but it's fixed on one MID. And then in the one-dimensional convolution layer, we have a kernel in a row shape that goes through different MIDs, and this captures the sum of isotopologs to be in some value. And then after these convolution layers, we have a fully connected neural network that can capture the training parameters to predict the ratio of de novo GMP synthesis to the overall GMP synthesis. And so we then evaluated our model on training, validation, and test data set, and we saw the similar correct prediction rate in training data set and in test data set. And this means that our model doesn't overfit, and it's generalizable on unseen data. Then we did an experiment on mice. So again, we have GBM PDXs and some GBM PDXs with the MMF treatment. So these two conditions, we have the 13C glucose tracing in these mice. And because this drug targets the de novo GMP synthesis, we expect to see a lower rate of de novo GMP synthesis in the MMF treated mice. And our prediction also shows this, because we can see that here the control condition has higher levels of de novo GMP synthesis, but the MMF treated one, it predicts that with a lower levels of de novo GMP synthesis. And then we use our model to predict the de novo GMP synthesis ratio in patients. So here you can see that we have these two non-enhancing and enhancing tumor samples in patients, and we can see our model suggests a higher prediction of de novo GMP synthesis in non-enhancing tumors. And we think that might be correct because the enhancing has more blood vessels in it, and it can take up the nutrients from the blood and circulation, but non-enhancing doesn't have any access to the nutrients in circulation, so it needs to synthesize by itself. And so that's why the de novo GMP synthesis is higher in the non-enhancing tumors. And in future, we'd like to further validate our model based on, again, mice data, but this time we'd like to have a time course experiment. And with multiple time points, we again can use our previous metabolic flux analysis model to find different fluxes and the ratio of de novo GMP synthesis. And we can also use our machine learning model to predict the ratio, and then we compare these two. Also we have a single cell RNA-seq data from these patients, and we'd like to find the fluxes based on the flux balance analysis and the gene expression. and then compare it with our machine learning prediction. And at the end, we'd like to use this algorithm in the clinical study. So this algorithm can predict which patient should take this treatment, and the MMF would be beneficial for them. With that, I'd like to thank my advisor, Dr. Deepak Nagras, and Dr. Anjali Mittal, and all the people in my lab, and also our collaborator, Dr. Daniel Wall and Dr. Andrew Scott. They did all these experiments, and they gave us very helpful feedback. This is the end of my presentation. I'd be happy to.
This is one question. I understand that you only had that was not sufficient to give predictions for your standards to utilize the models. Okay. This is more of a general question. For a non-steady state model, how many data points would you want to have before you feel confident that you can plot the model?
Yeah, so for time course experiment, we have 4 time points, I think at 30 minutes, 1 hour, 2 hour, and 4 hour. And the surgery of patients, it takes about between 2 to 4 hours. So we would like to design our experiment to be in that range.
Okay. So it's based on the how long the great hypothesis and then 3 data points for the events, and then another data point for all cases.
Yeah.
Thank you.
Sure.
Anyone has questions online, you can put them in the Q&A box. And if anyone else in the room has questions, of course, please feel free to raise your hand.
Yeah.
In the CNN model, why you use the 2 convolution layer that you can Yes.
Yeah.
Well, the first layer you using the convolution 2D, and why you like to using the convolution 1D, or you just directly flattened it? The convolution 2D and then skip the convolution 1D and directly to the flattened layer.
I see. Yeah. I think the flattened layer doesn't have any trainable parameter, but convolution layer has some. And I think by adding this, I could see like about like 30% increase in our correct prediction rate. So convolution layer has some trainable parameters. But the flattened layer doesn't have any trainable parameters.
Yes,
so so if we just remove the convolution 1D layer, we reduce the model in training side. So we eliminate some of the training parameters in convolution 1D layer, right? If we eliminate that layer.
Okay, thanks. And the 1D means that you convolution it in the y-axis.
In y-axis?
Yes.
Here? Yeah.
No, the x-axis.
X-axis?
Yes.
Right.
So I'm wondering the meanings about the convolution 1D. How you convolution it? Convolute it.
I see. So it's like a row vector. So when we want to define the kernel size, we just have like 1 dimension for the kernel, and that's why it's called convolution 1D. So we basically have like some of our, some metabolites do not have any 13C carbon in them. And the other metabolites may have like 1 carbon or 2 or 3, 4, or 5. Here I have it and plus 1 to 5. So this is the number of 13C carbon in a molecule, right? So when we have like this convolution, it actually captures all these 13C carbons in these metabolites. And the rest are the carbon 12, and we are not interested in them because we just use the 13C glucose to find the enrichment patterns, right? Did I answer your question?
Okay, okay.
Are there any other questions? I don't see any questions online either, so we can thank our speaker one more time. Coming and hope to see you all next week.